Okay.
All right, let's take a look. We're going to continue with the extracellular matrix. We looked at the ECM before, and as we said last time around, the extracellular matrix is secreted proteins and polysaccharides. One of their major functions is to glue cells together to form tissues. In other cases, they can form parts of external skeletons. Think of cell walls of plants. They can form arthropod exoskeletons. They can also form internal parts. Think of bones, ligaments, tendons, and stuff like that. That's all extracellular matrix produced by living cells within these types of tissues. So that's one function. And of course, the other thing, things like cell walls, protect cells from osmotic stress, protect cells mechanically, and things like that. We described some of the polysaccharides and some of the proteins. The major proteins, including the collagens and the fibronectins, are perhaps the most significant and abundant in these proteins, but we mentioned a few others and stuff as well. But the extracellular matrix also has another important kind of hidden function. It acts like a signaling agent. And the reason why is it activates through a different route the very same RAS-MAPK pathway that many growth factors activate. So in other words, it means that the extracellular matrix can stimulate cell differentiation and cell division. Now a little history in this. Here again, people trying to culture human cells. They had a very hard time. They found out that you had to add blood serum containing all these growth factors, which they didn't know what they were in those days. And even then, the only cells that they could grow in culture were cells that came from the nastiest, aggressive cancer that you could find. Ordinary, non-cancerous cell, you throw them in a big bottle with all the growth factors and stuff, and nothing would happen. The cells wouldn't divide, their metabolism would be very low, and eventually they die. Now somebody came up with the idea that, hey, in a body, these cells are constantly crawling around and they're constantly in contact with this extracellular material that surrounds each cell. So maybe there's something with it that these cells need. So they decided, let's make a fake extracellular matrix. So they got some purified collagens, some fibronectins, all that kind of goodies, and coated the bottom of these culture dishes with the stuff. Put the cells in, and lo and behold, they started to act normally. They would attach to the bottom of the dish, or at least to the ECM on the bottom of the dish. They would spread out like a fried egg, and they start crawling around. And most importantly, their metabolism would go up to normal levels. They would divide actively. And that's what we do most of the time when we're culturing human or animal cells. You have to do that. The ones that don't require ECM are basically usually very aggressive cancer cells. So that says something. That says that this material, these big polysaccharides and proteins on the outside of the cell is affecting cell function. It's changing the cytoskeletal because cells are moving and crawling around when they didn't before. It's stimulating cell division. And if you take embryonic cells and put it in the right kind of extracellular matrix surface, they will differentiate normally. So, that's telling us something. Now, the cell does not take this material inside. It stays on the outside. What is affecting something going on inside the cell? That means there must be some kind of signaling. The extracellular matrix must have a signaling function. Binding up to the ECM on the outside stimulates changes on the inside of the cell. So let's take a look at that. Now there's a whole bunch of different things that the extracellular matrix can do. So we're going to have to look at them kind of one by one because there's no way I can stick all that stuff on the blackboard at one shot. Okay, but let's take a look here. All right, here's a cell membrane. Now outside there's lots of ECM. I'm just representing a bunch of fibers. Now the first part of any cell signaling pathway, what's the first thing you need in the cell signaling pathway? You've got to have receptors that bind something, some kind of ligand or whatever. Okay, so that means there must be receptors for the extracellular matrix. And lo and behold, we have a big diverse family of them. And they're called the integrins. I'm going to put a couple down here. The integrins are extracellular matrix receptors. Okay, the integrins are a pretty diverse family. There's a number of different genes and stuff. And integrins, what they do is there's two subunits. You can mix and match. So you can have one kind of integrin sticking to another kind and so on. The integrins are huge proteins. They got molecular weights in the hundreds of thousands. They go completely through the membrane. Of course, all receptors have to do that. And each individual integrin has multiple binding sites for different extracellular matrix components. Some will bind certain types of collagens or certain types of fibronectins or certain types of polysaccharides, and others won't. But each integrin has its own selectivity of what kind of molecules it will bind. Now, of course, when you bind, the integrins bind to the appropriate extracellular matrix. Joe, question?
I'm trying to read what's behind binds there.
Each binds multiple ECM proteins or polysaccharides. Okay, now, when a receptor binds to whatever its ligand is, some extracellular matrix component, the receptor undergoes a structural change. That's things that receptors have to do, and that activates things inside the cell. First of all, let's take a look at the signaling function. We're going to be dealing with both signaling through the integrins and altering the cytoskeleton through the integrins. But we're going to have to do those separately just for space consideration. So first, let's look at the signaling. What's going to happen here is when the integrins will activate two types of kinases. Now, I don't think it's a direct binding, I think it's an indirect thing, but there's two different kinds of kinases. One is called Sarc, S-R-C, and the other called FAK. I get in trouble if I just... Okay. And they're both protein kinases with numerous targets when they're activated. FAK stands for something called focal adhesion kinase. We'll see what a focal adhesion is in a few minutes. So it's another kind of kinase. These two kinases get activated. Now they have a number of different targets, but one of the biggest things they do is when we turn these guys on, they eventually can phosphorylate and activate, among other things, a guanine nucleotide exchange factor. These are the proteins that are going to stimulate GDP for GTP exchange in G protein. They tend to activate G proteins. And this one happens to activate our old friend, the G protein RAS. That's the same one we see in the RAS-MAPK pathway. So, out goes the old worn-out GDP, in comes a fresh GTP, and once again our covalently bound lipid anchoring into the membrane. So now we have active RAS with GTP, and it's active alpha-steroid. And then the rest of the pathway continues. We activate the RAF kinases, and they phosphorylate and activate our MAPKK kinases. And then those guys phosphorylate our third set of kinases, the MAPPKs. And it's these guys, the MAPKs, that go out and phosphorylate all kinds of things, stimulating cell division, turning genes on and off, which can stimulate cell differentiation, and so on. So, in effect, the extracellular matrix acts as a solid-state growth factor. Now, we're finding in embryonic development many times what will stimulate one type of tissue or one cell type to differentiate is either the release of soluble growth factors like our EGFs or FGFs or what have you. But not always. sometimes what does it is being in contact with the right extracellular matrix. Now, the wrong extracellular matrix won't do anything. But if you have the right extracellular matrix composite, the kind that those set of integrins bind to, then you can get cell differentiation. I'm going to give you one example since some of you folks coming in are going to take 413 in the spring. There are these cells called the Crest cells. Now what happens is early in embryonic development you have this structure called a neural tube forming. It's a hollow tube that runs the length of the embryo, anterior to posterior. That's going to be your central nervous system, your brain and spinal cord. As that neural tube closes, now in humans this is like 2 weeks into development, it's really early on, it's one of the earliest steps. As that tube closes, some of the cells at the very top of the neural tube, instead of becoming part of it, sort of migrate away, and those are what we call the Crest cells. Now these Crest cells spread all over the embryo and settle down in all kinds of different parts of the embryo, proliferate, and then differentiate into things. Some of them become endocrine glands. Some of them become your jawbone. Some of them become your peripheral nervous system. Some of them become the pigment cells or melanocytes in your skin. So they can turn into a whole bunch of different cell types. Now the interesting thing is, if you take Crest cells from an embryo that are in one location, it's supposed to become a jawbone, and you stick them somewhere else, they'll turn into something else. It turns out that what causes these cells to turn into specific cell types is the extracellular matrix in the neighborhood that they move into. If you alter that, they will turn into something else. So these transplant experiments show that the location where those cells settle, which actually turns out to be differences in extracellular matrix in those areas, that's what causes them to turn into specific cell types. So in other words, their fate kind of depends on what neighborhood they grow up in. Now once again, Crest cells, unfortunately, when they start going bad, Crest derivatives, when they start going bad, turning cancerous, they tend to be some of the nastiest, most aggressive, cut you down fast kinds of cancers. Because what they do is kind of revert back to what they did in their childhood. They spread all over the place and proliferate. Think of melanomas. Think of gliomas. That's the glial cells that are support cells in the central nervous system. That's what cut down Ted Kennedy a few years ago. Those are really nasty, hard to treat, aggressive cancers. Those Crest derivatives are nasty when they go bad on you. Okay, well anyway, so we're seeing here, what's going on is we're activating the RAS-MAPK pathway. So not surprisingly, the right extracellular matrix acts as a growth factor because it fires off the same pathway. Now just a little historical interlude with this guy here. Sarc. Sarc stands short for sarcoma. Sarcomas are soft tissue tumors. They're cancers. Now, go back in the 1970s. You had, when Nixon was the president, he decided, he made a big public thing, we're going to have a war on cancer. We're going to try to come up with, cures for cancer and stuff. So he created this thing, part of the National Institutes of Health, called the NCI, National Cancer Institute. Now, unfortunately, he didn't put any more money into it, so what was happening is the NCI was getting money from the rest of the NIH budget. It didn't increase the budget at all. But in those days, there was a some prominent people were also involved in the NCI, thought that most cancers were caused by viruses. Now, some are, but most of them are not. Okay, so somebody known this virus called the room sarcoma virus, or RSV. And what would happen, it infected things like chickens and ducks, fortunately not humans. But what would happen with RSV is it would cause these soft tissue tumors that we call sarcomas in them. So it's a cancer-causing virus. Now, around that time, early 70s, they just started developing the techniques of molecular biology and gene splicing. So it's like, oh, we have a cancer-causing virus. Let's see what kind of genes it has and what those genes do. Well, look at this virus, and they find it only has 4 genes in it. So they successfully knock out these genes one by one. 3 of the 4, if you took any one of those out, the virus simply could not infect cells and replicate. But if you remove the 4th gene, the virus would infect cells, but it would not cause cancer. It would spread, it would infect the cells, go through its life cycle and stuff. spread to other cells, but it wouldn't cause cancer. So that one missing gene was a cancer-causing gene, or what they called an oncogene, the first one discovered. And they called that gene SRC, Sarc, for sarcoma, because that's what it caused. Now, the next question is, what does this gene do? What does this protein product do? Well, around 1974 or 75 or so, one of the hot-button labs in this thing decided, hey, we're going to figure out what this gene does. So what we're going to do is splice in the ordinary lab bacteria E. coli and have that bacteria produce a whole bunch of this stuff and see what this stuff is. And they mentioned their plans at a big conference, a big scientific conference. And then people said, whoa, step back for a minute. Okay, you are planning to take a known cancer-causing gene and stick it into a bacteria that is a normal inhabitant of our digestive tract. Is this such a good idea? So that started this whole thing about regulation and stuff and potential safety risks with genetic engineering, with gene splicing and stuff. And as a matter of fact, there was a moratorium by the scientists called saying, let's step back and not do any kind of gene splicing experiments until we have a better idea of what kinds of risks we're dealing with. The federal government got it, set some rules. Now, since then, a whole bunch of other tests said, the risks are much less than what was originally anticipated. So almost all molecular biology experiments, you just do an ordinary microbiology lab. We've got people here doing that. There's no special facility. Only occasionally for things like deliberately splicing pathogen genes and stuff into bacteria, you have to have higher containment. So the risks are a lot less than they originally anticipated. But anyway, for a while, I caused a whole bunch of it. Well, the question is, of course, eventually that experiment was done, by the way, but in much more controlled conditions. And the idea is, what is Sarc? Well, they found it was a protein kinase. Not only that, it was a kinase that phosphorylated proteins on tyrosines, a tyrosine protein kinase, the very first one discovered. So they said, my God, we have a cancer-causing gene that sticks phosphates on tyrosines, which is something they've never seen before. So they started getting interested, looking at any kind of tyrosine protein kinase because of its potential links to cancer. And then they asked the question, okay, we got this viral gene that sticks, that's a kinase that sticks phosphates on tyrosines, the target proteins. Now, do we have anything like that in our own genome? And using molecular biology techniques, they came up with the answer, yes, we do. But the viral version of Sarc was a little bit short, and its kinase activity was continuously switched on. The cellular version, a little bit longer, could be regulated. Remember, protein regulation, very important. Okay, so that kind of led to the thing of what Sarc was, and led to a whole bunch of interest in tyrosine protein kinase, and many of them involved in signaling pathways, and when they go bad, they can push a cell to cancer state. We already described some of those. Well, the other thing is, what's involved in it? It took a long time. Eventually realized it was part of the connection between the extracellular matrix and the RAS-MAPK pathway. So that's kind of where we stand at this point. Okay, that's what we see here when we're dealing with the signaling aspects of the extracellular matrix. But the ECM does more. Because we mentioned it stimulates cells to change their shape, to sprawl out and attach to the surface. It stimulates cell movement. So that means somehow we must be dealing with the cytoskeleton, the cell structural framework, effectively the cell's bone and muscles. So let's take a look at that.